Hello, everyone. Welcome to our second session of the year of the Signals Connect Community Series. Today, we are going to take a look at Signals Synergy, a solution for streamlining CRO collaboration and information exchange between pharmaceutical and biotech sponsors with their CRO partners. When added to Signals Notebook or Signals One, Signals Synergy overcomes challenges in communication, planning, and information exchange, eliminating the errors, wasted resources, and delays created by traditional systems. And if you were listening carefully, you may have noticed that I mentioned Signals One, and you may have thought, what is that? We've made some exciting enhancements to Signals Research Suite, and we thought it was important enough to rebrand Signals Research Suite to Signals One. All your data, simply all in one, with AI-ready data for today, tomorrow, and beyond. You'll be hearing more about Signals One starting at BioIT World 2025. But since you tuned in today, you're getting the exciting news a few weeks early. My name's Chris Stump, and I'm a product marketing manager for Signals One and for Signals Synergy at Revity Signals. And I will be your moderator for this session. We have a fantastic webinar for you today, but before I introduce my co-presenter, I want to share what you will learn today in today's webinar. So what are you going to learn today? Well, today we want to talk about pharma and biotech challenges when dealing with the CRO partners and how we have developed a solution for that collaboration challenge. So today we're going to cover key challenges in the sponsor and CRO collaboration within drug discovery, the 5 primary capabilities of Signal Synergy for streamlining sponsor and CRO collaboration, and give you a closer look at the software, including upcoming new enhancements. I already introduced myself, and now it gives me great pleasure to introduce my co-presenter, Heather Hayes. Heather is our Signal Synergy product leader. Heather has a PhD in biomedical engineering from Georgia Institute of Technology. She's been doing a great job of translating sponsor and CRO collaboration challenges into a really great product that I think you're going to like. Before I jump into the presentation, I have 2 housekeeping reminders. First, this webinar is being recorded, and the recording will be shared with all attendees in the next couple of days. Second, we plan to have Q&A at the end, so during the presentation, feel free to add your questions to the Q&A box. So without further ado, let's jump into the presentation. And most of what we will show you today is available in the product today. We will discuss the future direction of the product also, and Heather will clearly indicate that as such. Okay, so I'd like to start out by talking about the key challenges facing pharma and biotech. The first of those is the increasing complexity of drug discovery research. So over the past 15 to 20 years, researchers have been working with a broader spectrum of molecules, ranging from small traditional molecules to nucleic acids, peptides, microbiomes, and stem cells. These diverse drug modalities are not only harder to represent, harder to make, and harder to deliver, necessitating greater collaboration across scientific disciplines. The complexity is further amplified due to the intricate nature of the biology involved. To manage this complexity calls for a next-generation discovery informatics solution. And one approach to managing the complexity of drug development from the biopharmaceutical industry perspective is really outsourcing. So that's setting aside, a need for software. This strategy is particularly useful when in-house capabilities are lacking or when there's a need to reduce cost and improve operational efficiency. Outsourcing to contract resource organizations has been a practice since the 1940s, initially for clinical trials. This expanded in the 1990s to include intermediates and building blocks, and over the last 15 to 20 years, there's been a significant focus on drug discovery. CROs offer deep expertise in various drug modalities and access to cutting-edge technology, which may not be available in-house. Thus, outsourcing becomes a viable option. Outsourcing to get access to cutting-edge science and cost efficiencies from CROs in China and other regions has been the norm for the past 15 or 20 years, but that may change. As many of you may already know, the U.S. House of Representatives passed the BioSecure Act with bipartisan support. If it had been enacted, it would bar U.S. companies from receiving federal funding if they work with CROs in certain countries. While not an outright ban, it would be an effective ban on certain CRO partnerships. Although it seemed likely to pass last year in the Senate, it is currently on hold. Additionally, a new presidential administration has issued several executive orders affecting NIH funding and tariffs, creating further uncertainty about partnerships. So given these factors, scientific complexity and business uncertainty, a next-generation informatics solution also needs to enable nimble collaboration and rationalize data sharing because sponsors are going to need to be able to pivot between partners, and despite the differences between CRO partners and sponsors, sponsors are going to need to be able to efficiently exchange data with their CRO partners. So just why does a next generation informatics solution need to help simplify complex scientific research and enable flexible collaboration? Let's kind of take a look at kind of the high level kind of challenge. So specifically at the challenges facing pharma and biotech sponsors that are working with their CRO partners. So CRO collaborations are predominantly run using spreadsheets and email. Sponsors request assays via manually maintained spreadsheets. They communicate back highly complex scientific data in Excel-based reports, often generated or consumed with copy and paste. It's incredibly inefficient and error-prone, and in other words, costly, resulting in wasted time and money. We held extensive interviews with our customer advisory board to understand the specific workflow challenges, and I want to share a few of those with you next. And I really appreciate this slide because it gets to the heart of the key challenges. So allow me to walk you through these. So starting at the top on the left-hand side there. So a small biotech reported receiving Excel reports in varying formats from their CROs, requiring reformatting they lack the staff to handle. So this often requires someone with programming and data wrangling skills. The second bubble there, another biotech noted up to 25% of the reports they were getting back from their CRO had errors in there, and that necessitated sending those reports back to the CRO to be corrected. And then on the third bubble, pharma companies often work with many CROs, and it's cumbersome to kind of manage all the user accounts because they often give access to some of the software systems that they use, and it's really hard to understand like when users come and go at the CRO because it's an external organization. And then finally, the last bubble there, another pharma company highlighted the difficulty in tracking project status and knowing when data is ready for use due to the sheer number of projects that they have going on. So our solution, Signal Synergy, addresses 5 key customer pain points, and I highlighted some of those. And the first one is providing simplified user administration, and Heather's going to talk more about these and kind of show you what this looks like. An area for drug design, so an ideation workspace for capturing drug designs and hypotheses, with built-in IP protection, so built-in masking of proprietary codes, properties, and material IDs to protect intellectual property, scientifically minded project management, and rationalized data exchange, so quickly being able to consume and utilize the information being generated at the CRO. This slide gives you a bird's eye view of what some of this looks like. And on the left-hand side, in the black border, you see the interface for establishing and managing collaborations. The middle screen in the yellow border shows the idea space where researchers can manage structure ideas before deciding to send to their CRO to be made. And finally, on the right, in the red border, is a project management Kanban board that shows the status of the work orders, which is a great tool for understanding the status of ongoing projects. And Heather is going to give you a deeper look in a moment. So Signal Synergy leverages the Revvity Signals technology platform that is a cloud-native SaaS platform. The platform is built on ChemDraw, the gold standard in chemical communication, and Spotfire for advanced analytics and visualization. We leverage this platform to deliver specific industry solutions to address the scientific complexity that drug discovery researchers are dealing with, as well as enabling nimble collaboration. Today, we will focus on Signal Synergy for streamlining CRO collaboration between pharma and biotech sponsors with their CRO partners. And as you can see from this diagram, Signal Synergy unlocks CRO capabilities in Signals Notebook and Signals One. And as I mentioned in the introduction, we're rebranding Signals Research Suite to Signals One, and you'll begin hearing more about the exciting news starting at BioIT World 2025, and where we'll be unveiling some powerful new capabilities. So when Signal Synergy is added to either Signals Notebook or Signals One, it overcomes challenges in communication, planning, and information exchange, as I highlighted earlier. So let's take a deeper look as I turn the presentation over to Heather Hayes. Heather, the floor is yours.
Thank you, Chris, for that introduction. As Chris mentioned, I'll take you through a tour of the product. We're going to start with the administrative side, carry you all the way through the sponsor experience and the CRO experience, and help you see how the fit for purpose Tooling and workflows of Synergy can really alleviate some of the biggest challenges of sponsor CRO collaboration. Also effective for non-CROs, whether that's CDMOs, academic partners, any kind of external partner that you're working with. So as Chris mentioned, one of the biggest challenges that sponsors face is administering many collaborations with multiple CROs. Each one requires security setup, user management, establishing the right group associations, and also managing the users within each one. The high turnover of CRO scientists only further compounds the problem. So one of the goals of Synergy is to allow you to easily pivot between CROs and adjust your reliance on different CROs based on your evolving business needs and the ever-evolving international environment. So to reduce administrative burden, Synergy provides a CRO repository for easy CRO onboarding and offboarding. Each CRO is automatically ring-fenced as soon as you add them, meaning that a CRO can only see what belongs to them and their projects, only what you intentionally share with them. Rather than requiring complex security, including new groups, group associations, roles, security policy, it's quite straightforward to add a CRO with just a click of a button and know that they will only see their work and what is explicitly shared with them. In addition, within each CRO, there are subgroups. We call them departments. And so if you need to, you can have a collaboration that only involves some of those departments and not everyone in the CRO. By treating CRO users as a distinct type of user, we can also ensure that no proprietary data is unintentionally leaked and sponsors always maintain full control of external work. Optionally, each CRO can have its own CRO administrator. This is completely optional depending on your business rules. A CRO administrator can help manage their own groups based on the number of licenses that you've offered to them and help organize them into different groups or departments that might perform specific work, while of course the sponsor always retains oversight over the licensing, users, and what the CRO can see. Finally, Synergy provides flexible licensing allocation for CRO users to allow the sponsor to easily adjust the distribution of users, which CROs you're working with, and easily offboard a given CRO based on the dynamic business needs. After setting up a CRO in Synergy, the sponsor can easily initiate a collaboration with a CRO. A collaboration creates a dedicated secured slice of your tenant for a CRO to exchange work or experimental records, data reports for a specific drug discovery project, for example. A collaboration is assigned to internal user groups. These are the groups of sponsor users or internal users that are going to oversee the work. And again, they always retain full control of the work and experiments. And then you can add individual subgroups or departments within a given CRO that need access to this collaboration. And this can be dynamic over the course of the collaboration. Maybe initially you're working with some medicinal chemists and later you're working with screening or a different group. So this can be dynamic as you need. You can easily bring these on and off the collaboration. Importantly, we know that our customers heavily rely on attributes or fields on their experiments, notebooks, and collaborations to help organize, secure, and search for their work. However, some of these attributes may be proprietary, such as maybe there's a new Alzheimer's disease target or a top secret project code, or anything that you might need to organize all the work by, but you don't want the CRO to know. The sponsor can easily hide these fields from the CRO simply by marking them as hidden from CRO, while maintaining the ability to search by them, organize their work, and see this metadata on the sponsor side. Again, because the CRO user is now a unique type of user within Synergy, we can automatically hide this proprietary information. These fields are never visible to the CRO in the UI, search, APIs, but on the sponsor side, they remain usable in advanced search and always remain visible. So the sponsor can use the same attributes that they always use on their experiments and notebooks on all of their external work, bring those together in one database, easily search and organize them while protecting all of their IP and proprietary information. So where does a collaboration typically begin? It often begins with an idea and the process of evaluating and ideating and maybe even synthesizing potential drug designs. So ideas are a new entity in Synergy for capturing and tracking sponsored drug ideas. Each idea you can think of as representing a hypothesis about how to make or improve a drug. And inside an idea, sponsors can propose designs in little sets that we call design tables. They can propose these designs for small molecules or biologics. And these designs are like sketches on the back of a notebook of proposed structures or sequences. They are truly distinct from registered materials. They've not been made. They don't necessarily have an amount or a known appearance. And as you know, many of them will be successfully synthesized, but many will not actually progress and lead to a true lead. So there's no need to clutter up your material registry, but you do need to have a record of who ideated them, when they were ideated. You need a place to bring together predictions. So to send out these structures and gather predictions, maybe through some 3D modeling and silico modeling, some computational tools, and gather these predictions and begin to easily prioritize these different designs. So that's what an idea represents. Synthesis tracking and tracking of the progress of these designs is a key strategic area for our product that has been the focus of our partnership with our early adopters, especially in early 2025. For our medicinal chemists, we are expanding the tools in Synergy to allow them to have designs across many statuses, all the way from a proposal state to when it's ready to be sent for synthesis, in synthesis, all the way through registration and synthesis complete, or a failure state when you close out that design. So even when you're sending out large sets of designs in a single work order, which we'll talk about later, they can know when each one is complete, in progress, or even place it on hold for a time if there's a problem. So we've added a Kanban board for at the individual design level, as well as a data grid view for our designs. So it's very easy for our medicinal chemists to work in this ideation and design space and know how things are progressing, whether they're being worked on in-house or outside. This is something that's coming very soon. This is going to be released in the next release or two here in spring of 2025. We want our chemists and our decision makers to say that now they have a place, and Synergy really expands the entire Signals platform to be design, make, test, all inside of the Signals platform. They can track the work being done on each lead and know which leads are truly progressing and are ready to go into preclinical work. So once you've got your leads, maybe they've been synthesized in-house, a design has been synthesized, or maybe you want to send that out for synthesis. If it's been synthesized in-house, it may be time to send out some batches for screening or work, or maybe begin to scale up. That's where work orders come in. Work orders are a secure way to share select information with the CRO. Work orders are prioritizable. They can be group assigned to a CRO department. Maybe you don't know exactly who's going to work on it, but you know which group is going to work on it. Work orders offer an expanded set of statuses to keep both the sponsor and CRO automatically up to date on the progress of the work. Again, no need to have emails or other tools, maybe a spreadsheet with a list of assay requests. No need for that. This can all be done inside the Signals platform. Work orders can contain documents, chemical drawings, tables, anything you might usually use in the Signals platform can be added into the work order to communicate what is needed. You could include your SOP or instructions or tables of information. So again, no more digging around by the CRO through their emails or spreadsheets to find what they need to work on. It's all right there within Signals. Importantly, work orders are fulfilled by a sponsor-controlled and templated Synergy experiment. So you can set up what type of information you want back about a given screening assay, or what type of information you need them to include for a synthesis work order, and make sure it's all properly captured, and importantly, has those attributes, even if they're hidden from CRO. So this gives the sponsor control of the format in which you want the data back, and then the CRO can deliver as many of those as they need. One of the most important features of a work order is the ability to control what is passed to a CRO. In general Signals, sharing operates at an entity level. So if you can both see the same experiment or the same material, you both see everything. Synergy is uniquely designed for working with external partners, so that even when you share an entity like a material or a design with a CRO, you can choose to have them only see certain properties. So you can choose only to share certain properties of that design or material with the CRO, only the ones that they need. So when a material, batch, or design needs to be shared with the CRO, the sponsor can select which properties are shared to the CRO and which properties remain sponsor only in the reference table. That's a unique new kind of table inside of the work order. So you don't have to share the entire entity. This is super important to our customers because there are times when they need to share all of the secret sauce to maybe make a design, but maybe they don't want to know about some of the modeling or predictions that they've done on those designs. Or maybe you're sharing out some batches and shipping those batches over. You want to identify those batches, but not maybe keep the CRO scientists blinded while they're running the screening. So that's what reference tables allow you to do. So what does this look like when you're going to add designs or assets or batches to a work order in the reference table? The sponsor is offered a list of properties that are determined by the admin and marked as shareable to the CRO. For a given work order, they can select which ones they want to share for this particular work request. You'll also notice that some of these properties are marked as sensitive with this little triangle. This means that the admins configured these to require an extra privilege. So you can have certain persons on the sponsor side that can share sensitive properties and certain ones that can't, just adding that extra layer of security. Ultimately, the selected properties and only the selected properties are then shared in the reference table for the work order, protecting that IP while also providing the CRO with everything that they need. In the case of materials, we know that sometimes one of the most sensitive or proprietary pieces of information is actually the material ID, as these are often used in patents, publications, or submissions. So in Synergy, we provide an external ID in addition to the material or batch ID for all the materials that you're using with your external collaborator. So you can use this external ID to identify the batch or asset with the CRO while protecting or hiding your material ID. Any data that's reported back, you'll see both the compound ID and this aliased untraceable EXID, whereas the CRO is only working with that EXID. Important, these EXIDs don't allow batch traceability. So if you send out batch 1 to the CRO at one time and later send out batch 10, they won't be able to trace that back to the same asset and link up that data. So this is a screenshot of what the sponsor sees with both the compound ID or the compound batch ID and the EXID, as well as which properties are shared to the CRO, whereas the CRO only ever sees the EXID and the properties. We imagine our customers experience benefits such as the sponsor project manager, Synergy allows me to protect my material IDs and choose when to share the compound structure or protein sequence. I can share as much or as little as I need based on the work. And as a CRO scientist, I have everything I need within the work order. I sign into Synergy, I land right on my work queue. I know what's in progress, what's coming up, so I can plan my work. It makes it very easy for the CRO as well. Work orders also come with powerful user-friendly project management tools like a Kanban-style work queue for both the sponsor and the CRO. Each work order has a work type that helps you easily identify, oh, these green ones are related to synthesis. The orange ones may be more screening related. These work types allow you to easily identify where maybe you are in a given collaboration or with a given CRO and how each work is progressed. You're able to filter these down to maybe just a CRO group you're looking for, or maybe one person you're working with, or maybe only the work orders that you've sent out. It makes it easy to track the progress of all the work through the scientific pipeline from synthesis to screening to preclinical animal studies. The CRO sees a very similar board that allows them to plan for what's coming up for them, plan out their resources, and also easily click in and see the experiments that they're working on in relation to this work and have everything they need in one place, all within the platform. This is where they're going to see the instructions, communicate with you via comments or within the work order, and then also deliver the results. So no more need for them to have Excel lists or email. Everything is right there for them. So let's walk through this experience. What does it look like for a CRO to come in? The CRO lands right on their workboard and they can navigate easily to a work order that they have in progress. Inside, they can see what you've requested them, maybe some instructions or particular formats you want back. Here, they've shared a set of designs and the structures that it's requested to make. Just as an example, they could also share some maybe intermediates and the EXID used to identify them. They can then click into their experiment. Within their experiment, they can add all the types of content that you're used to. So they could add documents, they could deliver PDFs, They could add some of their research. You can also include instructions directly into the template if you'd like, or as you're working together on the document, you can communicate here or using the comments. They can draw up maybe the design that you've asked them to synthesize. It's very easy for them using the quick add toolbar to bring over structures from the work order, such as designs. And then they can create samples that are then ready for you to receive and register. In a given sample, we have a new field called the work order reference where they can identify, sample 19 or sample 20. Oh, this is actually the physical sample that I made for design 93 as an example. And it's very easy for you to see, okay, these samples relate to which designs. And then they can easily sign and submit. You can set up different workflows for the CRO versus the sponsor. So you may require them to have a reviewer before they submit or have some signing procedures there. You can easily distinguish the workflows between the CRO and the sponsor. Now, when the sponsor receives it back, just as normal, they can register their samples. And when they register, it writes back the compound ID. But you'll notice that on the CRO side, they are never able to see that compound information or hover or click out to this material. They only see the external ID that's been assigned. Again, that's an untraceable alias ID that can't trace even back to the asset. So you can register from right within the CRO's experiment without exposing your material IDs or any additional information that you've added onto that material during registration. So the sponsor sees it all with links, and the CRO only sees what you've shared with them, what they've made, and the external ID if you choose to register. So what if the CRO needs to fulfill a work order with a series of experiments? This might happen because the sponsors requested synthesis of a large design set, or maybe it requires multi-step chemistry, or maybe an assay scientist at the CRO needs to rerun an assay. It came back, it had edge effects, and they need to rerun that assay. It's very easy for the CRO to create as many experiments as they need using this little green button at the top to create more experiments. Each of those are created in the template that you've specified and with all the metadata that you've specified. And then this table is populated, the synergy experiment table is populated to show you the progress, who created each experiment, if they're working collaboratively, maybe within the chemistry group at the CRO. They can see which ones are active, when all the experiments have been signed and submitted. So it's very easy for the sponsor to track the progress and link out to the work that's being done. You can even relate each experiment. Let's say you've sent a set of designs or a set of batches to be screened. You can easily relate each experiment to the given design that's being synthesized. Maybe there's 3 experiments per design because it's multi-step. So it's very easy to see, okay, well, this design has been submitted back and this is the data related to that design in this experiment. So it's a very flexible system no matter what the CRO needs to deliver to complete the work. And importantly, as I mentioned before, this concept of hidden from CRO fields, you as the sponsor, when you issue the work order, can set up hidden from CRO fields that might contain a project code or maybe a disease target that you want all of their work to be marked with, and keep it hidden from CRO. So even as they create new experiments, they will never know those properties are there, but all of their experiments will have that metadata on it for search. So you aren't relying on them to remember or to select the right project, but rather you can put those on there and keep them hidden from the CRO, never to be seen by them, to make sure you're organizing your data. These attributes are shared across the entire Signals platform. So you as a sponsor, when you come into an advanced search, even let's say over in the notebook, you can easily pull back your experiments and the synergy experiments or your external experiments coming from the CRO, find maybe all the active ones that have the project code, this Alzheimer's project code. So you can pull these all back. Everything is stored in the same database. Everything is harmonized with the same rational properties and the same powerful Elasticsearch. So again, this is harmonization of internal and external data. So a final piece of very powerful functionality provided in Synergy is often when you're requesting assays or studies from a CRO, they will return these beautifully human readable, but very complex and difficult to consume reports. They're often these Excel-based reports that are multi-tabular that have all the information about the work that they've done for you. These are usually very standardized from a CRO. So if you request a WuXi particular assay, for example, or from Pharmaron, sending whatever CRO you might be working with, they come back in particular formats. The details might vary across the format. So whatever tool we use to handle these reports needs to be able to handle some variability. For example, variable number of compounds or test items that are used, varying number of time points, case by case. Often these reports have multiple tabs. Maybe it's tested across multiple species. So one report might have one species, one might have a multiple. You'll often see merge cells, possibly with key conditions, in this case, maybe with or without an inhibitor. And you need to know that all this data relates to this condition of with or without an inhibitor. And finally, the raw data must all be connected to all the metadata that might be in some early header sheets or summary sheets about who ran the assay, maybe what the lowest level of quantification is. So you might also see some information about units in headers. All this makes it very difficult to consume these type of reports. So wouldn't it be nice if we could take this type of unstructured multi-tabular report and automatically convert it into structured data? This is exactly what the Synergy Data Exchange does. All part of the Synergy project. We provide a large, robust library of CRO report transformations that know these types of report that are CRO and assay specific, know these formats, and can automatically extract both the endpoint data as well as the well by well or individual measurement data. These are the report formats that the CRO always uses. It's their own formats. So we're avoiding them copying and pasting things from their own internal processes into maybe a specific load sheet. Rather, we're allowing them to deliver their typical report, saving them time and reducing the error that's often introduced in that copy and paste process, while providing you with the needed structured data. Once a report is uploaded, we automatically do some data validation to make sure that all the expected tables and required data that are there, and provide feedback to the CRO to let them know if something's missing from the report in a way that they can easily regenerate the report and upload it to correct those errors. So like I said, the CRO is driven by this library of transformations. So when the CRO provides the report, the data is automatically transferred and transformed into structured data with feedback directly to the CRO user, as well as documentation about what is expected. And from there, it's very easy to publish into Inventa or a data factory, or maybe even a third-party data lake if that's what you choose. For the sponsor, this means that simple data extraction, clean, harmonized data can be brought together with your internal data, which is really what's vital for unlocking the power of AI or machine learning and unleashing the power of the entire Signals platform. For the CRO, the data exchange means it allows them to deliver their typical reports, your assay or study reports, without the need to copy and paste, without the risk of introducing data, and improves their efficiencies and reduces their chance for errors. So just a quick demo of what this experience is like. Again, this is a CRO user going into a work order where the sponsor has requested a specific type of report. Here you see maybe the samples that are shipped over with those external identifiers, maybe requested test species that you might need it to be run on. And then when they click into their experiment, you'll see that the data exchange section has been set up for them with an expected report type. They can double check using this documentation button what's expected of them. They should recognize this as their expected reports, gives them some instructions. You can set up these instructions however you like. We provide an out-of-the-box set of instructions. You can provide some examples, de-identified examples. We provide some of those to you. You could even provide a template that they could download to fill out, although usually these are set up to be auto-generated from their internal processes and connected to their instruments. You can see that they'll recognize this as the typical reports that they usually deliver. So once they've reviewed that documentation, all they have to do is easily drag in their Excel-based report, and it will provide them with some feedback, whether that be friendly errors or a validation to know that this report is an accepted format. You'll notice that they only see the report and they don't see the results that might include metadata that you may not want them to see, or the transform material IDs. On the sponsor side, they can see that, oh, now this work order has been submitted. They can filter these down to find that submitted work order. They also receive a notification. They can come in and then review the data that was delivered to them. You'll see that not only do they see the report that was delivered, which again, that report is likely in those external identifiers or some kind of CRO identifier. And if you're using our external IDs, we can automatically transform this data. So you see all the data is delivered from the CRO in the format of the EXID. But when you come to the results tables, which are the structured tables that come out, you'll see that all the data has been transformed from that EXID into the compound batch ID that's ready to merge with the rest of your data in Inventa or a data factory. You can see we provide a summary table of the endpoints as well as these what we might call well-level details or individual measurements. And you can easily download those straight to CSV or coming very soon directly publish those without ever leaving the experiment into Data Factory or Inventa. So in summary, Synergy really addresses 5 key customer pain points and really unleashes the power of the whole Signals platform for external work. It provides simplified administration of CROs and CRO users with a CRO database and the ability to dynamically adjust your CRO ecosystem, IP protection with automatic ring fencing of CROs and the ability to protect at the sub-entity level metadata or other IP and make sure that that is never leaked, even if you make a mistake on the administration side. Synergy also allows the sponsor to manage and track their drug designs for a complete design, make, test, decide cycle in the Signals platform. Project management tools like Kanban boards, not only for work orders, but also for designs without ever leaving Signals or relying on spreadsheets to manage work requests. And finally, the data exchange automatically extracts structured data from CRO assay and study reports that allows for harmonization of external and internal data for downstream machine learning, AI, and decisioning.